As you know, was telling, this is going to be the topic of my application talk. So just an overview. So what we did in this work, we were using CHO cells, Chinese hamster ovary cells. We were transfecting or co-transfecting those cells with subunits of potassium channel. They were fluorescently tagged. And then to study their dynamics and oligomerization state, we were using a turf microscopy. So we were getting the illumination and signal only from the membrane. And our time lapse was then analyzed with three different fluctuation techniques that probably you heard about a lot during this workshop. So they're going to be IMSD, number and brightness, and 2D per correlation. A brief introduction of the voltage-gated potassium channel. They are formed by 4 alpha subunits. Each one has a voltage sensing domain, and they are regulated by 1 or more beta subunits. Some of these channels, for example, the one that we're interested in, KCNQ1, are constitutively active, addressing membrane potentials, and also have a role, for example, in epithelial cells. The complex that we're interested into, that is KCNQ1, that is the main subunit, and KCNE2, that is one of the beta subunits from the KCNE family, is an epithelial complex that is essential for normal gastric, thyroid, and choroid plexus function. And we were trying to address some question about the molecular composition and the subunit dynamics of this channel. So the first step for us, so this is a collaboration, I forgot to mention, between our labs, so between me, Michelle, Enrico, and Lorenzo, that you heard that talked yesterday about the spectral phaser part, and Professor Abbott, still at UCI. So in their lab, they proved that the fluorescent tagging of our subunits that will be either MEGFP or mTERI don't alter the channel functionality. So their lab did some measurements with whole cell patch clamps, and they saw that the currents generated by the tagged version of KCNQ1 show similar properties to the one that normally you can see when the KCNQ1 is not tagged. Also, they saw that the tagged version of the KCNQ1, KCNE2 were constantly active, and they were expressing a small occurrence compared to the KCNQ1 alone. And that was similar to what they were observing with the corresponding untagged version of the channels. So once we made sure that our system wasn't altered for the physiology part, we moved to our part of measurements. So as I mentioned before, we were using a turf microscope. So, and for some of our sample, we were either using the GFP tagging or the mTERI tagging. So our system have 2 excitations. And then, as I was mentioning, since we're looking at proteins in the membrane, we were using the turf mode where only a single plane is illuminated. So we were collecting the signal coming essentially only from the membrane. And then our signal were detected and then split into 2 channels, and we were using a CMOS camera. Okay, so if you remember, we were saying that our data set will be analyzed with the 3 different techniques. So the first one is IMSD, image mean square displacement. These techniques, just a brief summary, can use the spatiotemporal fluorescent fluctuation caused by the protein diffusions to obtain a picture of the protein mobility in the nano environment. In particular, from the equation, you can extrapolate different interesting parameters for the protein dynamic. For example, the size of the domains in which a protein is diffusing, length of confinement, the diffusion coefficient inside these domains, the micro, and across the domains, macro, and also the mode of diffusion. In particular, we can see here a schematic representation of the free mode of diffusion, so the protein can go anywhere, the confined mode, so there's the strong domains, or the transiently confined. Applying that to all of our samples, so either the channels, the subunit, the beta subunit transfected alone, or the alpha subunit transfected alone, or the co-transfection, we could see that for all of these conditions, the best model applied is the transiently confined mode of diffusion in the membrane, independently if the subunits were expressed alone or co-expressed. Going into the details of the parameters that we could obtain from this analysis, we first noticed that analyzing the microdiffusion, we saw that when E2 was co-transfected with Q1, we could see two distinct populations. So we decided to split our distribution in two, and we were calling this one like the slow E2 Q1 population, and this is the fast E2 Q1 population. So as you can see from here, the E2 slow population has a similar behavior if we're looking at the macro and the length of confinement compared to the E2 when transfected alone. While if we're looking, for example, at the length of confinement, the E2 fast population was displaying a similar behavior to the Q1 or the Q1 when co-expressed with E2. So our conclusion from that was that the E2 fast population was probably the one that was binding with the Q1, while the E2 population represent the unbound population. We also tested if there was some dependency from these parameters to the expression level. And here we base that on the intensity, at least for the range of intensities, the for expression level that we were considering, we didn't see any dependence on the expression levels for any of these parameters, no matter if the proteins were singularly expressed or co-expressed. Okay. So that was our finding for MSD, the first kind of analysis that we did. Then we perform another kind of analysis, that is the 2D percorrelation function. So just briefly, this is an important kind of analysis that measures the directionality of the protein diffusion. So with this kind of analysis, we can build a map of diffusion for the entire image by cross-correlating the pixel as a function of time and in different angular direction. So by seeing the deformation of the percorrelation function, we can somehow see if the protein is facing an obstacle. So, and we can discriminate, therefore, between an isotropic diffusion, where we can use eccentricity as a parameter. So if the protein has an isotropic diffusion, the eccentricity is going more towards zero, where if you have obstacles, the eccentricity is going higher. So we have an isotropic kind of diffusion. For this kind of analysis for our project, we carried out this analysis at different percorrelation distances. So here you can see our results. So we calculated the 2D PCF function at 5 different distances. Here you can see the corresponding nanometer distances for all of our samples. We can see that KCNQ1 in green, whether a singularly expressed or co-expressed, show a low eccentricity, that is comparable with our inert control, that is like a membrane-bound form of MEGFP. Then we saw that for the E subunit, they show, especially at longer distance, a higher eccentricity. And they show either the E2 alone or the two population of the E2 co-expressed a similar behavior. If we are going at the short correlation distance, we can see that E2 has a stronger eccentricity, E2 fast, sorry, showing a stronger eccentricity. That means that E2 fast is localizing in different membrane domains where they have like a more pronounced motion directionality. And here you can see an examples of the map that you can build based on the eccentricity values. So we can see, for example, that overall, if you increase the per correlation distance, you have like a higher eccentricity for all of the cases, but then you can see differences in the behavior. And this is a comparison with intensities. The last technique that we applied on the same data set is number and brightness. So this kind of analysis calculates the average variance of intensity distribution for each pixel and is able to determine the concentration, so therefore the number of the proteins and their aggregation state. In this case, it is mandatory to use a reference of a protein that is known to be a monomer. So in this case, as I was mentioned, we were using the monomeric version of EGFP with the variance that is bound to the membrane to have a better reference. Here you can see our results. So for KCNE2, we proved that it exists as dimer when it's expressed alone. And then for our co-expression, we see that the fast population is in the form of a tetramer, while the slow population is, you can see here that is between a dimer and a tetramer. So since the trimeric version is not physiologically possible for this kind of channel, we interpreted that as a mixture of dimer and tetramers. While here, you can see that for the Q1, either if alone or co-expressed, they exist as a tetramer, and that is confirmed by other studies in the literature. Okay, and here, as I mentioned, we have our monomeric control for the GAP EGFP. Okay. Since I was mentioning that with this kind of analysis, we're using the same data set and we're applying the 3 different kind of analysis on the exact same sample, we can also try to understand better, for example, a correlation between what we see in the 2D PCF map and what we see in the map of brightness, and therefore the aggregation state. So just an example here, you can see in this region, here you have higher brightness region, and here you also have a corresponding strong motion directionality from the 2D PCF. While, for example, here, there's another region with a strong eccentricity, but that doesn't correspond to higher brightness. So this kind of analysis can be important to study in general membrane proteins and try to correlate the differences in different points of the membranes. So then we decided, since we have like so many parameters and so many data, to try to create a representation, in this case, the spider plot, to show the combination of the parameters that we can obtain with this multiplex analysis. And finally, we try to build a visual model for the behavior of this complex. So in particularly, here you can see that for all of the conditions, So going from the beta subunit alone or the alpha subunit alone or the co-transfections. You can see here that the confinement is represented by these square domains, and the size of this is the same as the size of confinement for each of the conditions. Then we were representing the micro and macro diffusion with these circles, respectively the dark blue and the light blue ones. And the diameters was the distance, average distance in this 2 time. The directionality that we can get from the 2D PCF was represented by these 2 ellipses, where the eccentricity of the inner one is the eccentricity from the short range to the PCF, and the outer one is the eccentricity of the large range, the largest range that we consider for the 2D PCF. Finally, here, there's a zoom in. We were trying to represent the aggregation state that we were obtaining from the number and brightness analysis. So here, as I was mentioning, we have E2, that is a dimer, Q1, that is a tetramer, and when they are interacting, they're most likely having like a 4-to-4 aggregation. So to conclude this, as I was mentioning, we found that this channel exists as an octamer with a 4-to-4 ratio. We studied the dynamic of the singular subunit and the co-transfected subunit, and all of these findings can improve the understanding of the physiology of this channel. And from the methodology part, I would like to highlight that the use of the turf imaging that is really used for membrane studies can be done in combination with all of these techniques. So from the same data set, you can get information about the oligomerization state for NMB, the diffusion modality for MSD, and the nano-environment organization. So this is an approach that can be easily applied to a large variety of different kind of studies.